So we saw how the electron transport chain is used to pump protons from the matrix into the intermembrane space. And that's called, and the energy involved in that proton gradient is referred to as the proton motive force. And we saw how that proton motive force can be used to make ATP. But there's other things within the mitochondria that use the energy of the proton motive force. For example, the transport of ADP and inorganic phosphate both depend on proton motive force. ADP is getting ADP into the matrix is dependent on the actual electrical gradient. Right? If you have a lot of positively charged protons in the intermembrane space, then you have a relative positive charge out here and a negative charge here. So anything, any sort of transport that moves a net negative charge towards the intermembrane space is going to be exergonic. And that's what this adenine nucleotide translocase takes advantage of. It moves ATP that's being generated in the matrix, it moves it out, and in so doing allows ADP to move in. Now that's a change of a negative charge moving out, and so that's exergonic, and that's what fuels these reactions, these movements here. Now, the phosphate translocase symporter is a way to pump phosphate groups into the matrix. And the way that works is it takes advantage of exactly the proton motive force. Protons move down their concentration gradient through this symporter, and they drag along with them phosphate groups, thus pumping phosphate groups into the matrix, and then they are able to be turned into ATP. Brown fat is another interesting tissue, and it's a tissue that's located within newborn mammals, hibernating animals. It's brown because it contains a ton of cytochromes. But the important fact of brown fat is it has another protein within the mitochondrial inner membrane that other tissues, the cells of other tissues, don't have. And it's called an uncoupling protein, or UCP1, or also called thermogenin. And all it is, it's a proton channel. It offers another way for protons to get through, go down their concentration gradient and get into the matrix instead of going through ATP synthase. Now, when it goes through ATP synthase, it releases energy, and that energy is used to make ATP. When it goes through this proton channel, this thermogenin here, it releases energy as well, but that energy is not being used to, it's not coupled to anything. It's not used to be doing work. So all that energy gets wasted as heat, but that's exactly what the animal wants. It's used to keep newborn mammals warm, and it's used to keep hibernating animals warm. Just that simple, protons moving down their concentration gradient, releasing energy, and that energy being released as heat just over and over and over again is sort of a heat source for these animals. Now, of course, their ATP production in these cells will be a little less efficient than normal, but the upside is this heat source. So these are other ways in which the cell is using the energy inherent within this proton motive force to do work, to get things done within the cell.